So welcome to Ask the Expert, a brief, informative, and lively discussion with experts in type 1 diabetes and related interdisciplinary research. We're recording the event. We're going to post it on the Sugar Science site YouTube channel shortly after the presentation. If you have any questions for our guests, please feel free to enter them in the chat or raise your hand at the end of the presentation. And today we have as our guest, Dr. Austin Lefebvre, coming from UC Irvine. I understand he's a newly minted PhD, so congratulations. And I'm going to also share that he and his mentor and PI in the laboratory, Dr. Michelle Digman, are here with us. They have released a new Nature Methods paper, August 2021, which is pretty exciting. It's automated segmentation and tracking of mitochondria in live cell time-lapse images. And they introduce a tool called the mitometer. It's an algorithm for fast, unbiased, and automated segmentation and tracking of mitochondria in live cell 2D and 3D time-lapse images. And it's really kind of interesting. They use this mitometer in context of triple negative breast cancer and showed that the mitochondria actually move faster. They're more directional and more elongated than those in their receptor positive counterparts. So why are we interested in this? Well, because in type 1 diabetes, there's been a lot of discussion about what is the role of mitochondria in beta cells, during the dysfunction of the beta cell in autoimmune diabetes. And so this seems like it could be a really interesting tool to explore that. So welcome, Austin. Would you like to, give us a little introduction about about your work?
Yeah, absolutely. So thank you so much, Monica. That's actually, I think, the first time anyone's introduced me as a doctor. So thank you for that.
That's great.
So I'm going to try basically today to kind of talk about the work that I've done in graduate school, the main focus of it being on, this phenotypic investigation of mitochondria in cancer, but also extending that research kind of to how these organelles might be dysregulated or cause dysregulation in diabetes. And so maybe I can get started here on some intro slides. So, as I'm sure everyone here knows, diabetes can lead to dysregulated glucose levels, and actually hyperglycemia can lead to oxidative stress within the cells. And so, this oxidative stress can cause a lot of damage in cells. It can damage organelles and eventually, lead to cell death. And mitochondria are primarily known as obviously this powerhouse of the cell, right? Regulating metabolism and generating the energy that the cell needs to function, which in itself, dysregulation can lead to diseases like cancer or diabetes or obesity. But another crucial feature that the mitochondria are responsible for is generating oxidative stress via reactive oxygen species. And this process actually gets upregulated in times of hyperglycemia. Additionally, mitochondria are very dynamic organelles, changing their shape, size, and location within the cell. And are also able to, split apart and come back together, known as, mitochondrial fission and fusion. And the dynamics of these mitochondria and how they kind of, move around the cells and generate ROS and in a spatio-temporally relevant manner has been shown to be, very important, but remains kind of understudied in this field. And I'd say that the main reason for this, kind of gap in research is the lack of analysis methods for mitochondrial dynamics. So we take some examples, for example, one of kind of tracking mitochondrial movement in cancer cells. And we look at their methods section. We see that, they say time-lapse sequences were imported. Were you guys able to see my screen?
Yes.
Okay.
It's perfect.
If you look at their methods section, it says time-lapse sequences were imported into Fiji, and individual mitochondria were manually tracked using the manual tracking plugin. Similarly, if you take another example of looking at mitochondrial length in neurons or neuronal precursor cells after mitosis, and look at their methods section, they say the freehand tool in Fiji was used to manually trace the maximum length of mitochondria fragment. So obviously, this mitochondrial research is very broadly implicated, but these methods to actually study this and to analyze this is lacking. And if we take a video of the first example, we can see these mitochondria moving around in these two different types of cells, and you can see that they're kind of tracking the mitochondria by these tracks here, kind of clicking on the mitochondria one frame at a time through the time lapse. And if you're doing an N of 100 for your study, and the video is 1,000 seconds at 0.1 frames per second, that's 10,000 clicks for a single condition. Not only that, but it's also going to be biased for specific mitochondria. So you might be following, mitochondria that are brighter or faster. And of course, it's going to be user dependent, right? So everybody's going to be analyzing these mitochondria in a different way. Mm. So we wanted to basically, create a method to do this in an automated way, in a bias-free way. And, we have, a bunch of different microscopes in our labs, I think close to 13 or 15 microscopes, something like that. And I wanted to look at a bunch of different conditions. And of course, these mitochondria can be different shapes, complex in shape and size, and how they move. And to be able to do that all automatically is, a challenge in itself, which is, I think, why these methods are so lacking in the first place. So essentially, for this tracking to be done, first you have to go through a segmentation process. So being able to essentially, take this image that we see here, remove some of the background. So you can see we find the background here, remove it from the original image, and then basically apply a series of Gaussian filters and thresholding to essentially isolate these mitochondria and be able to have them, separated from the background. And so once we have them in kind of this separated form, they're essentially what we can classify as objects that we can then track through our time series. And so, this process is a little more complicated to make everything automated, but this is kind of just a brief summary, and I'm happy to dive into detail if anyone is curious.
Really beautiful. I
Thank
mean,
you.
just to look at it, I mean, it's also full of data, but it's actually beautiful to look at.
Yeah. I think this is the example I use for the cell pretty much in every presentation, just because I think it's such a nice picture,
Yeah. It's so fascinating how dynamic they are,
Yeah, you can even see kind of in this corner here, they're kind of undergoing this splitting process, reattaching. Some of them are moving really fast, some slower. It's interesting how kind of complex these organelles are.
Yeah, no, it really is. And so you guys were able to and in is this in the triple negative breast cancer cells that we're looking at?
Yeah, so this is a triple negative breast cancer cell, I believe transduced with a mitochondrial marker, essentially.
Okay. And then so and how would you imagine using this in a either sort of pancreatic beta cells in culture, or even, I don't even know if you could get in vivo. I mean,
absolutely
potential?
Yeah, I mean, I think as long as you can, find a way to image these cells, it's possible to look at the mitochondria within them. So there are plenty of stains available. I think the most common one is TMRM, which is tetramethyl rhodamine ester. And essentially just it binds to within the mitochondria, so that basically you can first off visualize them, but also the fluorescence is going to be dependent on the potential of the mitochondria. So usually, when these mitochondria are a bit unhealthy, they start to release this TMRM and become less fluorescent. And so you can actually also kind of quantify the health of the mitochondria just by looking at this intensity. And of course, hopefully run it through kind of these algorithms to look at, the motion and the morphology of these mitochondria, and maybe relate that back to some, phenotypic problems that you see in type 1 diabetes. For example, maybe some necrosis or some apoptosis, things like that. And I think that's kind of what I was trying to do with cancer, is kind of look at how these phenotypically diverse mitochondria relate to kind of these more phenotypic cell-wide phenotypic diversity in cancer cells, and how maybe you can correlate those together to find some therapeutic targets down the line, whether it be metabolic or mitochondrial.
Yeah. I like how you guys basically, gave a big nod to the open science movement. You've got the Mitomer programs written in MATLAB, right? In the
Yeah.
MATLAB GUI Mitometer app and the corresponding source
Yeah.
code are available as supplementary software, and then you have it
Mhm.
all available freely on GitHub. So that's really awesome.
Yeah. Yeah, and
And
actually I can show kind of the GUI that I have.
yeah, that would be
I
great.
have a slide here. So yeah, I tried to really make it user friendly where, any kind of biologist that has, their image, either 2D or 3D, can really just, press these buttons and everything is kind of automated. Will show up, all of your images will show up either in the segmentation tab where you can take a look at the actual morphology of these mitochondria, in this tracking tab where you can take a look at, the actual tracks and a histogram distribution of, all your different parameters that you want to look at. If you want to compare different groups, so here I have, in blue, just kind of control cancer cells. And in orange here, these cancer cells treated with nicotazole, which is basically a microtubule depolymerization agent, which will slow down the mitochondria. And you can actually visualize that here. So this was, really an attempt to kind of make it as easy as possible for any user, not just those who are familiar with coding, or not just those who are familiar with, MATLAB or things like that. Really, anyone should be able to use it.
Yeah. No, that's
Mhm.
pretty amazing. And it's great that you have a very easy onboarding, it looks like anyways.
Mhm.
And so
Yeah. Mhm.
what's next for you guys in the laboratory?
Mhm. Yeah, that's a good question. So right now I'm still doing a few experiments on kind of how these mitochondria react to their environment. So we have these cells that are cultured in these complex ECM conditions, things like hyaluronic acid. So glioblastoma cells and hyaluronic acid gels, which is very kind of similar to their condition. These brain tumors in the brain, kind of similar environments.
Yeah.
We also have our breast cells in these 3D collagen and matrigel cultures, which are more similar to kind of the breast tumor environment in vivo. And we want to really see how, these cells are responding to their environment. So if we, target these pathways that are allowing these cells to contract, or allowing these cells to kind of sense their environment, how is that modulating the mitochondria? And how is that, kind of modulating their invasive potential downstream to kind of be able to metastasize?
Yeah.
I also add one of the advantages to this technique is that we can make use of additional type of imaging, for example, fluorescence lifetime imaging of the mitos, which is an autofluorescent feature that all mitochondria have due to their NADH amounts. So we can correlate metabolic states based on the lifetime of the NADH component within the mitochondria.
Mm-hmm. Yeah, and essentially this is a super powerful way to basically in vivo, in live cells, be able to say these cells or these mitochondria are upregulating oxidative phosphorylation versus their control counterparts, or vice versa.
Yeah, and you could
Um,
see how this could be very, very, this could be a key um, piece of data
mhm,
as you sort of track I guess, the dysfunction, the
mhm,
onset of dysfunction in beta cells.
exactly. But yeah, actually, I just have one more GUI slide that I
Oh
kind of
yeah.
wanted to show. But basically trying to make it, so you can test out these different parameters as easily as you want. So this is just kind of like advanced settings where you can kind of play around with it and see if something doesn't work like automatically, you can kind of tailor the features and try to make it work for your data set. But also, it supports batch analysis, so you can take in as many images as you want and just kind of let it run in the background. And is also compatible with ImageJ. So if you're more familiar with ImageJ, you can have that open and then import your images into Mitometer, and then back to ImageJ if you want to. And of
And what
course,
was
there's
oh,
a
sorry.
big, no problem.
No, I was just going to say, how long were you able to sort of track the cells?
Yeah, so I think the longest video I have right now is like 10 minutes.
Mhm.
But it's going to kind of depend on your cell and how stable your fluorophore is,
Yeah.
and how, I guess, constantly you're imaging. So if you're taking frames every half a second, you're most likely to kind of that fluorophore will probably start decaying, like not decaying, but not being able to fluoresce after some amount of time, quite a bit quicker than, if you were taking one image every 10 seconds, let's say.
And for these data, what was your image? What was
for these
the time
data,
frame?
I think it was on the order of two minutes per cell. And that seemed to be enough to really get, these strong significances and being able to visualize these strong differences between cell types. And that was every 1 second I was taking an image.
Okay, that's really interesting. And so if someone has, someone's really interested in the mitochondria, say, for instance, in some beta cells in culture or alpha cells in culture, any islet cells in culture, really, and they wanted to pursue this, should they just sort of reach out to you if they had like questions, or would you guys be interested in a collaboration? What are your thoughts on that?
Yeah, I mean, I'm always happy to help with, any problems that people run into with the software. And, as long as they can find someone, or, even if they're close by, come to us for like imaging of these cells, that's, I think, not a problem. Because it's really just kind of, as long as you can get these cells imaged with a mitochondrial label, you should be able to run this software no problem. So hopefully, basically, I tried to make that not so much of the bottleneck anymore.
Yeah.
Yeah, if that makes sense.
Well, it appears you succeeded. This is really a major, a major contribution with this methodology.
Yeah, I
And
appreciate that.
yeah, for sure. It's really, I mean, it's really, really great. And to get this sort of like out there in nature methods is a really a great feather in your cap as you go out into the postdoc world. Are there any questions from anyone else in the audience? You can feel free to throw something in the chat or just to unmute yourself and ask.
I have a question. I was wondering how this might do with something like islets that are multicellular and trying to see, something that is more than just one cell thick, I guess.
Yeah, sure. Great to see you, Rachel. It's been a while.
Yes, likewise.
So, I think with multicellular, it's fine as long as you can get a good enough resolution for your imaging. So I guess it depends on how you want to analyze your data. So for example, for all of my data collection, I tried to collect, a single cell in the field of focus and basically analyze the mitochondria there and then pull all of that data together to make kind of one group. But of course you can also do, like one big frame with a lot of different cells in plane of focus and just kind of average all the mitochondria within there. But obviously the only drawback with that is that you'll probably have to increase your resolution, which will increase your imaging time most of the time, and essentially kind of either slow down the time between frames, so you might not get as good of a temporal resolution, you might not get as good of a spatial resolution than if you kind of zoomed into one cell. But again, that's going to be all cell type dependent and really kind of depend on your system. So it will take a little bit of tailoring the microscopy aspect to your data set and to your cell type.
Yeah. Okay. Thanks.
Yeah. Thanks.
Is there anyone else who'd like to pose a question for Austin while we've got him here on the line? Okay, well, that was great. Austin, this is really just a great system. Thank you so much for sharing it with us and walking us through it. And I am hoping that we're going to introduce you to some folks that are over in Europe who are really quite interested in microscopy and tracking some of the organelles in beta cells. So I think I can't wait to hear what happens or what comes from those discussions, because I think there's something really powerful here. So thank you again. And I'm going to stop
thank you.
and thank you to Dr. Digman as well for joining us.